Okay. Welcome for another exciting seminar. I have the privilege of introducing Kevin Cox. Kevin is a HHMI Hannah Fellow at Washington University, and the Donald Danforth Plant Science Center. He's a recent hire. Kevin earned his bachelor's in biology at the University of Missouri, St. Louis in 2015. He then got his PhD in plant pathology at Texas A&M in Hugo Shan's lab in 2018, where he studied the molecular mechanism of a cotton disease called bacterial blight of cotton. Afterwards, he joined the Danforth Center for his postdoctoral experience in Blake Meyer's lab, where he focused on developing methods to study spatial gene expression of plants. Kevin started his role as a senior research scientist, principal investigator between WashU and the Danforth Center in January 2024. So very recent. He will be a joint faculty member as an assistant professor in biology at WashU and assistant member of PI at the Danforth Center starting in July 2024. So with that, let's welcome Kevin and learn about Into the Pond and Beyond, a journey into duckweed, transcriptomics using spatial and single cell biology.
Awesome, thanks for that introduction. Can everybody hear me? Okay, cool. And I'm gonna make sure I use this. So cool, thanks for that introduction. It's great to see everybody. So as Josh said, I'm a joint hire between WashU and the Danforth Center. And so what I basically gonna talk to you guys about before I get started. So before I get started, I'd like to introduce two things. So one, this is the first time I'm up here since the last time I was interviewing about less than a year ago. So there's a little bit of PTSD kind of sitting there. Very high stress like situation and just on this app stage. So that's one thing. And the second first is like, this is probably the first seminar I had to give that is on my birthday. So. I'll answer the question for you. I'm 33 years old, so make whatever judgment you want out of that. So anyway, so today I want to kind of talk to you guys about two stories. So one to share a little bit of background of like what I did in my postdoctoral work as far as like my training and how that led to me getting interested in spatial biology and single cell biology. And then, so that'll be like, have some data to that part. The second part to where it talks more about duckweed, that's something that I kind of picked up towards the end of my postdoc. So there's not much data there, but I want to kind of share what my plans are for my future lab going forward as I start to grow. Cool. So plant cells, we know they detect different stimuli, such as like environmental stresses, other stresses, whatever, using these plant cells that we all know and love. And so in order to regulate that information, what plant cells do, they detect whatever stimuli they can in that plant cell and relay it down to the rest of their organs, such as the leaves, the stem, and the tissue. And so in order to understand how these cells communicate and regulate information across like tissues and organs, it's kind of becoming important for us to understand like their spatial context or how these biomolecules are being communicated across the leaf or across the root or across a stem. And so that's where I got into this subject called spatial biology. And so you can kind of think of spatial biology as like the study of biomolecules, but studying them with their spatial context. And so a couple of examples here is that like you're now able to visualize where are certain genes or proteins are specifically expressed in tissue section with an SY coordinate. And so a comparison with that is that both in single cell sequencing can kind of show you the parts, but when you add spatial biology to it, you can now get a whole picture of like where stuff is located, where stuff is expressed at within an organ or a tissue. So that will be like the lecture definition, but I have family and I'm the only scientist in my family, and I had to normally explain what I do for a living. So what I like to do is I like to compare it to Legos. Everybody interacted with Legos, they either built them, stepped on them, whatever. So if you think of bulk sequencing or like bulk RNA sequencing or bulk sequencing in general, just think of a random pile of Legos. So you don't know what these pile of Legos make. It's just a random pile. You might look at this pile and say like, oh, you see a lot of blue ones, so those are obviously the more important ones, and us, the rest, maybe not so much. Or vice versa, you don't see as many, I don't know, white ones or brown ones, so those obviously cannot be that important. So it gives you like an average expression of genes or whatever biomolecules you're looking at, and you don't know what shape this set of Legos they make. So when you have spatial biology, now you've got the spatial context to it. Now you know where everything goes. Now you know what things are connected to. And you can start to say like, oh, some of the red Legos, they interact with not only some of the orange, but some of the pink ones as well. And you're like, okay, well, maybe it's a good idea. I didn't say the white ones or the brown ones, they were not as important because they're actually critical to make this whole piece work. And so just think of spatial biology as an idea to where like it kind of puts everything in a spatial context and kind of allows you to detect things at a higher resolution. So that's my definition I kind of share, or my analogy I kind of share with my family to kind of help them understand this topic. So I'm going to start off by sharing a little bit what I did for my postdoctoral work and kind of like my training and like how I got into this spatial biology area. And that'll be just basically 2 quick stories. So basically the research in my lab will basically use spatial biology techniques to kind of study different biological questions. And so for my postdoc, as Josh was saying in my introduction, I used that to kind of, I was using my postdoc more so as a method development to kind of develop methods to study different aspects of biology. And so one of these techniques I want to talk about is spatial transcriptomics. And so you can think of spatial transcriptomics as basically you add in spatial context to your gene expression. So you go from, let's say, taking a mouse brain to now know where that specific mRNA or your mRNA of interest is specifically expressed in that SY coordinate. And so this is not a novel technology. It's been around since the early, since 2016 in the biomedical field, and it kind of really took off over a few years recently. So this is a powerful technology to where like you can now take your RNA sequencing data and now add spatial context to it. And this, I just want to kind of highlight, there's 2 different methods to kind of understanding spatial, performing spatial transcriptomics. So one is a untargeted method and the other one is a targeted method. So the untargeted method is basically like an unbiased capture of RNA. So you capture like you would like a general RNA sequencing experiment, it's an unbiased capture. And you're now able to kind of figure out like where things are laid out as. On the other hand, the targeted is more kind of like hypothesis testing. So let's say, let's say the 5 or 10 genes you want to look at. You just want to make sure that they're right there. Well, with targeted, you just design specific probes that target those genes and kind of confirm that they're expressed in that spatial location. So the untargeted, I'm going to talk about that's a project I was kind of working on developing an approach for that. And most of that is like an array-based like spatial transcriptomics approach. And so to kind of walk you through how this process works is you'll basically have a plant leaf that you'll freeze and you'll basically take a plant leaf and freeze it. And what you'll do is you'll make these cryo sections to where you're making these very thin slices about like 10 to 15 microns thick and putting them on a glass slide. So on this glass slide is thousands and thousands of capture probes that have a poly-T capture tail. And that poly-T capture tail, that's what captures the poly-A mRNA. And the reason why you want to put this on the slide and take a picture of it is that way you have your map, that way you can refer back to it later after you get your sequencing results back. So you image it, you'll then release the mRNA through permeabilization. The mRNA will kind of be released, go down, get hybridized to the capture probes, and then you'll synthesize cDNA on those capture probes. And then once you do that, you'll remove all this tissue using enzymes, cell wall degrading enzymes, reactions, and then you'll cleave all these probes off, put them in a tube, and do your traditional library prep. And when you do your library prep, you'll basically get 2 different reads back. So the first read will be whatever mRNA or cDNA that you capture, or whatever transcript that you capture. The second read will be the spatial barcode sequence. And the cool thing about this approach is that each of these capture probes have a unique barcode sequence. So that means you're able to take whatever transcripts you capture and whatever barcode sequence you have and pretty much map where that came from. And what you end up is with a nice spatial expression data in 2D. So again, this technology is not novel. The drawback with this right now, especially for the plant biology, is that most of these technologies that are used for spatial transcriptomics, they're mostly for biomedical or mammalian systems. And you can see like some of the examples where like there's been a couple studies been published in plant systems, but for the most part, it's mainly done in mammalian systems. Additionally, these platforms tend to be very expensive. And so when you're talking about like a smaller like plant biology lab, it becomes very cost unfriendly to kind of go invest in these systems and do the spatial transcriptomics. And so I was kind of interested in developing a platform that was not only cost effective, but was also plant friendly as well. And to basically describe that, what that looks like is this is kind of still in its processing or like a development stage. But basically, I collaborated with this collaborator in Germany who made cDNA microarrays. And so we had these DNA microarrays synthesized on a NimbleGen platform, and we're able to synthesize not only high resolution microarrays that are about 10 microns resolution, but we're also able to synthesize these arrays at a very cheap manner. And so our vision for that is that like once we can establish this technology, we can kind of share it to the rest of the community, and people will be able to do spatial transcriptomics in plants for a very cost-effective, Okay, so that's spatial transcriptomics. So that part is still going on. So the next part that I'm kind of more excited to tell you guys is this technique called expansion microscopy, which is a side project that kind of developed like as I was interviewing. But it developed into something that's I got really excited about. So to take a step back, so we have all these fancy microscopes that can see these very high definition features, these molecules, these structures. So we can do that with these super resolution microscopy tools. There's some drawbacks with some super resolution microscopes. So for one, they tend to be very expensive. You have to be in a university or an institution that has quite a bit of money that can get the grants to purchase one of these machines. And so if you're a smaller school, you might not have access to a super resolution microscope. And if you do have access to one, they tend to be complex to use. And normally, you either have to spend hours and hours to get trained on that microscope, or you hire somebody that has like years and years of training on that microscope. And they're also slow to use. And what I mean by slow is that it's slow to collect the data, it's slow to process the data, it takes a lot of time to kind of go through and analyze that data. So instead of making microscopes that continue to analyze at a higher resolution, it would be easier if you could just simply make the cell bigger, right? And that's what expansion microscopy is. It's not sci-fi, it's actually something a group developed about 10 years ago at MIT. And so basically expansion microscopy, the way this works is you'll have these cells and you'll embed these cells in this polymer. And this polymer, what it does is that once you fix the cells and lock in the volatile molecules, you'll embed them in this hydrogel that's swallowed with water. And so once you put the cells in this hydrogel, you add water to it and it expands the gel about 4 to 5-fold three-dimensionally. And if you don't understand the chemistry to this, just think of baby diapers. So the group actually took the chemistry that's in baby diapers and basically applied it to expansion microscopy. And so when you think about it, baby diapers, when a baby urinates or goes to the bathroom, the diaper swells up and theoretically keeps everything in place.
Yeah.
Theoretically. I have 3 kids, so I change diapers like quite frequently. So it's my favorite example to use. But yeah, so the hydrogel in baby diapers, scientists at MIT basically took that knowledge and applied it to expand cells three-dimensionally. And so the cool thing about this is it's very common lab equipment you need, very inexpensive chemicals that you require. And so it gives you like super resolution like quality, but without the need for a microscope or without the costs and everything. So this has been done in many different organisms and tissue. And this is just like a small snapshot of what's been done. So you got zebrafish, C. elegans, E. coli, human tissues. You can name it. And so you guys might be thinking, okay, Kevin, you're a plant biologist, so what about plants? So what I'm showing here, this is basically it that has been done for plant systems. And with each of these studies, each of the authors, they mentioned that there's been some drawbacks with each of their studies. So with this one, the group, they basically isolated nuclei and chloroplasts, and then they performed expansion on that. The drawback with that the authors meant this is that when you isolate these nuclei and these organelles and then treat them with these very harsh chemicals, you tend to get them very distortion and they tend to get damaged in that process. Another group, they expanded arabidopsis, but they mentioned that it was about just a 1.3 fold expansion, and their goal for that was more immunostaining than like super resolution capture. There's been another study done in Chlamydomonas, which is a unicellular algae, and that, to my opinion, was a very nice expansion. There's another group that also did this in Arabidopsis roots. And in this study, they were able to get expansion in Arabidopsis root, but it was only limited to certain areas of the root. So you notice most of these studies, there's not a uniform expansion of leaf organs or leaf tissues. And that's what I kind of set out to do, was to make a method that could expand, like, let's say, a whole plant leaf, a whole plant root, or the meristem at a uniform manner. And probably like my predecessors before me, I ran into a wall. And I mean that both figuratively and literally. So plant cells, they have a cell wall, as most of you guys know. So the cell wall is designed to be stiff and keep stuff out and not allow stuff to get in easily. And because of that, when you try to do your chemistry for expansion microscopy, that chemistry cannot get in because it's kind of hard to get rid of that cell wall. So at the start, I was struggling to get this technology to work. And my collaborator, Kurt Zimmer at the Danforth Center, he basically suggested, well, why don't you start backwards first and then work your way forward? And what he meant by that is by using protoplasts. So for those that are not familiar with protoplasts, you can get these by basically using an enzyme cocktail that digests the cell wall completely. And you'll get these circular protoplasts. Now, granted, you lose out on that spatial information that you would, because now you got like a pool of like individual cells. But the cool thing about these protoplasts is that they retain all their subcellular properties. So you can still look at subcellular components using protoplast systems. And so that's what my group we were able to develop, was this protocol to perform expansion microscopy of plant protoplast system. Since you have to give everything a name, I'll call it EXPOSE for short. So I'll be referring this to EXPOSE throughout my talk. So we first were able to validate that this EXPOSE method does work in the Arabidopsis protoplast. And you can see the top panel, that's an unexpanded cell. The bottom one is a expanded cell after expansion microscopy. And we show that this expansion is really robust if you look at the non-expanded and the expanded. And the cool thing about this is that we're not only able to expand the cell size, but also different subcellular properties such as the nucleus. And so now we can see these features that were originally difficult to see at a very high, much higher resolution. We also developed antibody labelings for EXPOSE. And so our idea behind this was to develop something to where after you expose or expand these samples, that you can label, that you can do antibody labeling to look at endogenous proteins of interest. And so the two endogenous proteins we looked at were actin and mitochondrial. And I should have blew this up this time. But the cool thing about this is like my favorite picture, is that it looks like the unexpanded is practically nothing. There is stuff there, I promise you, it's just small and tiny. But once you expand it, it really opens up this matrix. You can kind of get into the mitochondrial features of the protoplast. And so this just shows an example to where we can look at endogenous proteins as well as a higher resolution. And then we also developed an in situ hybridization assay with these expanded cells. And so in situ allows you to basically visualize mRNA of interest. And so you can see here, we picked CAV1, which is a transcript that's expressed, and we can now see CAV1 expression at a much increased resolution. So the things we can do with EXPOSE, we can achieve robust expansion of protoplasts. We can visualize endogenous proteins in these expanded cells. And then we can do in situ hybridization to visualize mRNA. So we were curious to see if does Expose enable imaging of these cell components within organelles at a decreased resolution. And what I mean by that is like, are we able to look at transcription factors or different proteins in these organelles at a much higher resolution? So we had a grad, or we still do. There's a grad student in the lab that I was working in during my postdoc. She is interested in these two transcription factors that are involved in maize development. And so these two transcription factors, these are basic helix, loop helix, MS23 and MS32. When you knock either them out, the plant becomes male sterile, so they don't produce any maize anthers or any pollen. More importantly, these two transcription factors are known to interact and cause transcription. So we were curious to see if we were to transfect these two transcription factors in maize protoplasts, what would happen after we expanded the protoplast? So we found some interesting insights about these two transcription factors. So the first thing is that once we expanded, we first transfected the transcription factors individually. And once we transfect these protoplasts and then expanded them afterwards, we noticed that you can indeed see an increased resolution of their localization within the nucleus. And so that was pretty cool. But to our surprise, what we did next was we transfected them both at the same time within protoplasts, since they're known to directly interact with each other. And we just wanted to see what we would find. To our surprise, they completely changed their localization pattern when they transfect. So individual transfection versus combined transfection, they seem to change their localization pattern. And this just kind of gives you an example of what you can do with expansion microscopy. You can now get a better insight into resolution of how these transcripts these transcription factors or different proteins, how they're localized in the nucleus or any other subcellular component. So this is great toolkit for expansion microscopy and plant system. I'm actually working on a manuscript right now that hopefully I'll have a bioarchive this month. Drawback is it's only in protoplast. And depending on how you might feel, that might make you unhappy. You can't isolate protoplast from my plant species. That's true. It's not routine to isolate protoplast from every plant species. And some plant species, they're still working out that protocol. This is cool, but it's an organ like a leaf or root. I agree. When you have these protoplasts, like I stated earlier, you lose that spatial resolution or you lose that spatial knowledge because now you just got to pull cells within a tube. They don't count. Okay. That's some times where reviewers comment where like protoplasts, they don't technically count as a system, but okay. Or you could hate them. Believe it or not, like each of these comments I've either heard or have gotten at some point in my career, including the last one. So it goes back to the point to where I'm trying to get expansion microscopy to work in whole organs or in like whole tissues. And again, the problem is I have the cell wall that gets in the way of the anchoring chemistry. And to kind of leave you a little bit on a cliffhanger, I'll use an analogy of my favorite movie, the Spider-Man Across the Spider-Verse. So it left those that saw the movie kind of left us on a stay tuned for part 2 cliffhanger. So I'll leave you on the same cliffhanger as in stay tuned for part 2. And so basically we're able to expand like a intact Arabidopsis seedling leaf. So we're able to take a Arabidopsis seedling, expand the leaf and get a very nice uniform expansion. To the point to where the nucleus is about 70 microns in diameter. And to put that in comparison, like plant nuclei are about like 8 to 12 micron normally. So this was a very robust expansion, and we're kind of excited about spreading this over to whole plant tissues. And I need to add this up here. About a couple of weeks ago, there was another group that they also were able to get this to work in whole roots as well in Arabidopsis. But their expansion was also much more even and much more robust than the one that I had showed you in the previous slide. So there's a lot of traction that's picking up now in the plant field for expansion microscopy. Okay, so that was basically my work for my postdoc, was I was focused on method development. And so now as I transition to this role, I kind of want to go into answering more biological questions. And so one question is plant microbe interaction. So Josh said during my PhD, I'm a plant pathologist. I got my PhD in plant pathology. I love plant microbe interaction. My joke I say is that if it infects something, I'm willing to study it if it gets me money. So I love plant microbe interaction. I'm not going to talk about that today. I'm going to talk about the second question I'm interested in studying, which is in duckweed biology. So the first question I normally get when I talk about duckweed is, what is duckweed? From both family member and also other scientists, believe it or not. So the duckweed, they're basically the smallest flowering plants in the world. They grow in ponds. Ducks do eat them. That's why they call duckweed. And they're actually kind of classified as an invasive species because what ends up happening is that ducks, they'll go into ponds, they'll eat them, they'll get attached to the feathers, and the ducks, they'll fly to the next pond. And the duckweeds end up getting dropped off there. So they're actually kind of considered an invasive species. Take that what you may. And they actually, yes, they do flower in the right conditions, and they do produce seeds. But the condition they pretty much has to be like perfect conditions to get these flowers and get the seeds out of these flowers. So I'll explain what duckweed is, and the next question I get after that is, all right, why are you working on duckweed? And there's a couple reasons for that. So one, these duckweeds, they're known to be carbon sequesters and phytoremediators. So they grow in wastewaters. They're known to be really great phytoremediators to kind of absorb this agricultural runoff that's in these waters and kind of remediate the water. And additionally, they're also known to be carbon sequesters. So if you think about like global warming, they're good for storing carbon in their plant systems. And they also have potentially biofuel potential. They also have high protein and starch content. So believe it or not only animals eat them, but humans eat them as well. So they're definitely in diets in many parts of Thailand and other parts of the world. I personally have not tried duckweed yet, if that's what you're thinking, but that's on my to-do list. But they have a high protein and starch content. So there's people that are looking at this, including NASA, to where they're trying to use duckweed to kind of send the duckweeds to space for like better sources of food, since they have so much proteins in them. They reproduce really fast too. So think of it as a plant that can reproduce in less than 2 days, which to me, that blows my mind. It's kind of like almost looking at a green yeast in a way. And they're so small that they're also very easy to phenotype. And I'll kind of go into this later, this study that we were able to do by basically being able to phenotype these duckweed in an automated manner. So, like I said, I'm really interested in spatial biology. And now you have duckweed that comes along, and you're just kind of thinking like, okay, why are you including duckweed in this research lab? So our research goal is to kind of uncover the spatial organization of genes. So to figure out where are genes spatially located within a plant, in order to understand how these genes are regulated and how they design these networks and how they're regulated across plant organs and tissues. So if you think about it, duckweeds, they're very tiny. And so theoretically, you can potentially use this as a model to get like a global view of gene expressions of an entire plant at a cellular resolution. And that's something that has not been done before. And so that's where I'm kind of interested in using duckweed as a model system to kind of answer my research agenda of my lab. So I'll walk you through a couple of projects that our lab would end up doing to kind of introduce this to you. And so I'm really interested in basically phenotyping these duckweeds with different stresses and ultimately getting this very high resolution like transcriptomic data. So to walk you through each of these, one project that we have in mind is to do whole plant phenotyping and transcriptomics. And so in our study, what we're able to do is basically, since the duckweed are so tiny, you could load them up in these like 96 well or even 24 well or even 96 well tissue culture plate. You put them underneath a microscope that has like an automated platform. You do a time lapse, and then you could use PlantCV or any computational modeling to kind of map their growth or model their growth or quantify the growth over time, excuse me. And so what you're able to do with this is you can now let's say have different wells full of like different nutrient treatment. So let's say one system well, there's more, it's like a titration of like ammonia, for example. Or you can screen different lines of duckweeds in one well. And so this gives you like a more throughput manner to kind of phenotype this. So this is one thing that me and some collaborators we worked on establishing a couple years ago. And to show one example, yep, I knew that video was not gonna play, that's okay. To show one example, what we did was we did like a proof of concept example to where like we grew one set of duckweed without sucrose and one with sucrose. And they're not gonna play at the same time because I gotta do this manually, but we'll make this work. Yeah, so you got the one. No, you kind of get the picture. So you got the one growing without sucrose, it grows a little bit more slowly, versus the one with sucrose, it grows more robust and quickly. And so then you can kind of take that, do some computational modeling with it and kind of quantify the growth over time. And so this gives us a good opportunity to, or a good platform to kind of do whole plant phenotyping, but also transcriptomics as well. And so we can take the duckweed lines that are interested of us, they show that show an interest in phenotype and do more in-depth transcriptomics to that. And so afterwards, one thing I'm also interested in doing is giving them a much higher resolution of imaging for 3D reconstruction. And the reason why I want to do that is because I'm interested in taking this transcriptomic data and resolving it to 3D. And to do so, there's two ways you can go about doing that. So you could potentially take my spatial transcriptomic method and create a Z-stack. And basically what a Z-stack is you think of it like a bologna slicer, to where like you have like a tissue and you're making slice after slice after slice, and you're kind of stacking these slices together computationally to make a Z-stack to understand like how they're expressed in a three-dimensional manner. And so this is just like a preliminary experiment that I kind of did with one of the duckweed species to basically show that we're able to capture like mRNA from duckweed. And so in this experiment, what I basically did was it's like a cDNA footprinting experiment. And so the idea behind this is that you'll have your capture probe, and after you capture your mRNA, you'll add these fluorophores to reverse transcription, like as you're transcribing your cDNA. And so the idea behind this is that if you're able to capture your mRNA, you get like this footprint that lights up and it matches whatever you put on there. And that's what it basically shows. So that right here, you see the bright field image. This one is a microarray scan, like after we had did the experiment and hybridized the mRNA. And so this is potentially one way you can do this with duckweed is you can take these slabs over and over and make the stack. The problem with that is that you'll get these session distortion. So again, think of it like if you're slicing a bologna, like not every slice is going to be even, not every slice is going to be equal. You got all these air pockets in these duckweeds that kind of make it hard to kind of stack and make this Z-stack. And it's a computational challenge as well. So another idea that I had was that, okay, well, maybe instead of doing all that, we can take advantage of the technology at the Danforth Center and use these 3D imaging technology and combine it with our 2D transcriptomic data. And so at the Danforth Center, we have a scientist, Keith Duncan, who pretty much like runs our X-ray microscopy. And so the cool thing about this X-ray microscopy is you can capture these plant features that are really hard to capture in general, in 3D. And so you can capture different plant features such as the apical meristem, the buds, you can segment, cell segment like the inflorescence. So you can really capture these features in 3D. And so I had approached Keith with one of my favorite duckweed species, Wolffia, which is a very tiny duckweed. It's known to be the smallest flower plant in the world. And just asked him like, hey, could you do an X-ray scan over this duckweed species? And the answer is yes, you can. And so this kind of gives us an example to where we can now take this duckweed species, get this 3D imaging, and potentially start cell segmenting this data, and can combine it with the 2D spatial transcriptomic data whenever we get to that point. And this right here, I'll play this one time before I talk again. So basically what you're looking at is you're looking at basically a transfer session. So the duckweed is on its side and the camera is kind of moving this way. So I play it one time for you guys. Just have you look through it. Okay. And so what you notice actually going through this duckweed, you go through it, you see the daughter prawn and stuff pop out, and you'll see like this weird, I guess whatever the meristematic tissue to where it produces like daughter prawn duckweeds, is somewhere in the center. Not much is really known what goes on in meristem or what type of genes are involved in the meristem. And so that's an area of interest where I could potentially investigate like what transcripts are being expressed in that meristematic tissue. And again, the idea is that I would take my 2D data, which is facial transcriptomics, and this 3D X-ray microscopy data, and ultimately combine that using computational methods to express this or to illustrate this in 3D. And so the next thing that my lab will focus on is, of course, using spatial and single cell biology to kind of map out these transcripts and cell types. And the reason why I want to do that is because I'm very interested in understanding like how are these genes are expressed globally in the entire plant, and kind of understand like what type of genes are involved in like the organization of the meristem. And so basically our idea of this is that if we can understand like what's going on in duckweed, we can kind of translate that knowledge to other plants. So let's say how the plant grow quicker or have it produce more fruit at a higher rate. Just a couple of examples to kind of go from there. And last thing, of course, is functional genomics. So before Arabidopsis took over as a model plant, there was a really big push to use duckweed as like a model system. What stopped that is that Arabidopsis, you can transform it easily, or relatively easier. And that's where duckweed kind of fell short with that, because there was a lot of trouble with transforming duckweed. And so basically, a project in my lab is that we will do functional genomics with duckweed and establish like a reliable transformation system with duckweed. There's some protocols that are out there for duckweed transformation, but they tend to work. They don't tend to work with only that specific lab and not reproducible as easily. So the idea with this is that like once we establish this transformation system with duckweed, we can now do duckweed genetics, functional genomics, and figure out what are these genes or duckweeds are doing by knockout or overexpression. Okay, so the take-home message for my talk, if you able to get anything from it, is that you can use these spatial biology techniques to kind of understand gene activity with a spatial context. So instead of having this pile of Legos, you can now understand what these Legos are doing and where their Legos are located at. And because since you're now putting genes in space, you can visualize these transcripts and you can now start to develop these gene networks. And that can also lead you to identifying novel targets to study, and that can lead to engineering more stress-tolerant crops or more food production, etc. And then hopefully also convince you that the duckweeds, they're small plants, but they have huge potential. And they're also an interesting model to investigate this area of spatial biology. Okay, the last thing I'll talk about, because this was kind of requested like in my email invite, is some of my outreach efforts. So one thing I have a passion about doing is kind of sharing my science or my assignment for science to kids at elementary school or primary school. Mainly because like as I was growing up, I didn't know you could be a scientist. I thought that was a thing to where you had to be a super genius in order to do so. And so the reason why I kind of go out to these schools and kind of help kids with science lessons is just to show them like, hey, I look like you, you look like me, we can both be scientists one day. And one of the cool things that I started doing recently, so these 3 pictures, these were taken before COVID, like, before COVID, where I was going out to classrooms or different schools. So now after COVID, like, the Danforth Center, we have a good collaboration with the Jackie Joyner Kerr Center, which is out of East St. Louis. And they have an academy there to where basically the students, they can get bus there after school, they can do activities, they can do their homework, and they can also have a small academy there to where they go to school there from like kindergarten up to 4th or 5th grade. And so the cool thing about this is that quarterly, me and a couple of the lab mentors that were in my postdoc lab, we go out there, we teach them we teach them science lesson. We kind of share like the reason why we did was like we taught them how to do the scientific method. And we get them basically fast growing plants and they made their own questions. So like, if you add more water, what plant will grow quicker? You know, it kind of helps the kids to get to think about like, hey, I can actually be a scientist and this is what we do for a living. And the cool thing about this is that they're second and third graders. And so sometimes they send you cute little letters like this that you can kind of stick to your office. So yeah. Okay, and with that, I'd like to thank my former postdoc advisor, Blake Myers. Who's now at UC Davis, as most of you guys know. Also like to thank Lily O'Connor, who is a grad student here in the PMB program. And she's actually being posted in my lab space so she can finish out her dissertation work. Lily and Sarah, they both contributed to the expansion microscopy work that I shared with you. And Kirk and his team in the ABL, they contribute a lot to expansion, imaging, and just like a very fruitful collaboration. And then also Mark Samosa and his team for developing the microarrays for the spatial transcriptomic works. And then my funding sources for HHMI and NSF. And lastly, I'd like to thank you guys for attending and for watching and Danforth for giving me this cool opportunity to kind of start a lab and go after cool biological questions. So with that, I'll be happy to take any questions if there are any.
And over there.
Thanks.
Yep.
I have two questions. One is just a technical question, and that is in the expansion work. There's got to be some limitations of it, and I was curious, you know, sort of what are the limitations, and are they different for different plant species, different cell types?
Yeah, that's a great question. So I can think of a few right off the bat. So a couple of limitations is that like it's very hard to get uniform expansion out of every cell type. So it's kind of, so normally what happens is like when you get expansion, it's mostly, let's say, the mesophyll cell, like the outer cell that expands, at least in my preliminary data that I have showed with the expansion of the whole leaf. And so you have to kind of be, you have to treat the cell carefully to where like everything doesn't fall apart, but also enough to where your chemistry can kind of get into every cell and you get this uniform expansion. Another drawback with expansion microscopy is you can't do live cell imaging with it because the cells are fixed, so everything's kind of fixed and dead, so to speak. So those are a couple of drawbacks to it, and you have to be really careful with the chemistry as well because it's real easy to not get even expansion if you just expand the cell or expand it as much.
So my second question has to do with some of the things that a lot of us here are really interested in, and that is species-species interactions, particularly like in pathology, and then also in ecosystems where you have mycorrhizae. And particularly thinking about spatial interactions, the ability to visualize that is really exciting. And I just wondered if you had any comments on that.
Yeah, so that's definitely, I wanted to talk about that, but I kind of cut that out of interest of time and kind of focus on one thing. But that's actually an interest of mine is to kind of look at not only the plant pathogen interaction, but also the mycorrhiza. And speaking of duckweed, I have one slide. So duckweed, they have a microbiome. And so one thing I'm really interested in is there's microbiomes that can characterize, but there's no molecular studies done on this microbiome. So it's not known what these genes are doing or like how they're interacting with each other, especially at a species to species level. So that's an area of interest that I really want to investigate, or is how are these microbes are spatially residing within the host plant. So
That wasn't a planned question, by the way. We did have a slide.
I put this in the last minute. I'm like, you know what? I should throw that in there just in case somebody asks me that. So
hey, thanks for the nice talk. I had a question for someone who's working on a non-model single cell switch transcriptomics group. There's so much of the work being done is on human mice specific fish. What are some of the issues you're going to be managing to try to get started on non-model group for folks who are interested in getting started on it?
Yeah. Yeah, so I know with, so the drawback with, the reason why the plant field, they took a little while to get started with it is one, like plants, they have a cell wall, and you have like all these other components that are in plant system. That it makes it difficult to isolate single cells and make them happy compared to the biomedical or other biomedical fields where they don't have that problem. Another issue is that the biomedical field, they have marker genes well established. Not all plants, especially like the non-model plants, they have good marker genes. And the reason why you need marker genes, especially when you do single cell, is because you need to understand, you need something that kind of helps you sort the cells and kind of categorize what cell type this is. And so that's the drawback with single cell, is that you don't have a good marker gene, then it's kind of, you're kind of like shooting in the black box. With spatial, you don't need marker genes, but the drawback with spatial is that the resolution is not as great as transcriptomics, like, or as single cell transcriptomics. So nothing speaks to like sequencing the transcriptome of an entire individual cell. And so that's why our group and definitely other groups, what they're doing, they're combining the two. Is they're doing both spatial and they're doing single cell, because the spatial, it provides you a map at least to kind of understand like where stuff is located at. And the single cell or the single nuclei kind of help fill in that gaps. Yeah.
I was really interested in what you had to say about duckweed being a potential biofuel, and I wanted to hear more about that.
Yeah, so I wish I would have had a slide on that too. No. So yeah, so there's groups out, so the continent duckweed, like you can potentially like harvest, and I forgot what the makeup within the duckweed is. But within that duckweed, like there's potential to where you can kind of harvest the duckweed and extract whatever you need to produce these biofuels. And because you can produce these duckweeds like at a huge volume within these tanks or within these plants, you can collect these biofuels like pretty rapidly. Something that I know some of the companies are doing, they're trying to figure out ways to kind of engineer duckweed to make sure they reproduce, let's say, vaccines, or like maybe they produce like biofuels even better. So the potential is there, basically. And yeah.
Is there any work that you know of that's being done on it? Like, what's like the progress with that?
With the biofuel stuff, I don't know enough to give you an answer. I just know it's being done, so.
Hi, I have a technical question. So when you do the space transcriptome, do you put a whole leaf on it, or do you do like in the medical field, this really thin and slide on? Do you need to make sure it's a variety of cells?
Yeah, so that's why, so to answer your question, yes, you freeze them and then you slice them about like 10 to like, I think it's like 8 to maybe like 12 micron thick. And so you use that thickness because you want to make sure you get about at least one layer of cell, maybe 1.5 layers. So yeah, it's like similar to the biomedical fields where like you have to freeze and slice it very thin. There are spatial methods to where they do whole tissues, like in the photomap I had showed at the beginning of my slides, where like they basically took a whole root, embedded it, and then they did like spatial resolution over it. So, but most of the time it's kind of like you freeze it and then you section it. Yeah.
Question on the distortion of cells. So when you're expanding it, what is it an acceptable range for like whether you can study it because it's like too much stuff or it gets too distorted?
Yeah, that's a great question. So the one tricky thing about expansion, and it's an issue that I ran into early, is that when you expand these cells and you're not careful with the expansion of cells, they do get distorted, especially if you're not careful with the chemistry or like you do uncontrolled expansion. Most of the time when you do expansion microscopy correctly, most of the cells, they have very little distortion. You will find some cells that they get distorted a little bit, but for the most part, they kind of maintain their integrity.
can expansion microscopy be used with yeasts and with bacteria, things that have cell walls that aren't plant cell walls?
Yeah, so I know different bacteria has been done. I know for sure E. coli, and there's been other bacteria that's been used as well. I want to say that there's a study that's been done in yeast, but I'm not 100% sure as well. But definitely, there's been group that they did in the different microorganisms with this.
And those cell walls don't pose a problem in that case.
I guess not, because part of it is the composition of a cell wall in the micro is not similar to plant system. So you kind of have to every chemistry that you use is different from every different organism. And that kind of goes back to an earlier question to where like, does it matter between different plant species? And it does, because even within different plant species, the cell wall composition is completely different. So.
Any more questions? I will ask one. I don't know a lot about this area, but I have some duckweed in my aquarium. And they're so small, and they grow so quickly, they're really hard to get rid of. I'm curious if there's a signature of like the small size that shows up at the transcriptomeless level, or like on a more functional level. Like I would assume that they don't have a vascular system since they're so small, and maybe they do.
Yeah.
Yeah, can you just tell me a little bit about what the small size of them it affects what you're seeing when you study these guys?
Yeah, the small size of them, so I guess, are you asking like, what features within the transcriptome like makes them small, or like?
Yeah, is there some signature of being really small that you see?
Oh, okay. I don't know. No, yeah, I'm not too sure, if I'm being honest. Yeah.
Okay, well, let's see any more. If you have more questions, you can join us just across the hall for refreshments. And with that, let's give them a hand.